Okay, it's close enough to 1:30. All I have planned for today is to do chapter one. And chapter one is always an introductory chapter. Also, I did this today, and there is at least somewhat. Nope, that's not the one I want. That's the one I want. There is now at least somewhat of a schedule on Canvas. And all it is when the exams are going to be, my best guess. And so exam one is going to be more or less February 6th. Exam two is more or less March 4th. Exam three is more or less April 16th, and then exam four has to be like May the 1st. Final exam is going to be the week of May the 4th. And I put this warning on here. At the moment, as of January 20th, 2026, these are my best guesses as to approximately when the exams are going to be. As we get closer and closer and closer to the exam date, the actual calendar date of the exam will become much more firm and it will be set. Now, the chapters that are on each exam are not going to change. And so, exam one will cover chapters one through five, and exam two, six through nine, etc. Now, all that being said, here is chapter one. Now, I am using the fourth edition PowerPoints. And if you have the 2025 edition, and there are significant differences in between the fourth edition and the 2025 edition, I will download the new PowerPoints and start using them. But I really don't think there's too much, if any, difference between the fourth edition and the 2025 printing. And so this is an introduction to genetics. You can say that genetics is basically the study of heredity and variation. Now, you can argue that genetics is the underlying unifying discipline in all aspects of biology. And with an understanding of genetics, it lets you understand how life can exist at all the different levels of complexity that exists at. And the overview, the big thrust of genetics is to one, explain how traits get passed from parent to offspring, and two, show the relationship between genes and physical traits. And so it's going to show the relationship, or at least shed light on the relationship between the molecular makeup of an organism and the physical description of that organism. Now, genetic discoveries are growing fast. And for example, as of early 2025, it became, I don't want to say legal, it became the insurance companies agreed to cover if you have certain genetic illnesses. You can now undergo genetic operations that will edit your genome and alleviate the symptoms of that disease. Now, at the moment, it is extremely expensive. But as time goes forward, it will get cheaper and cheaper and cheaper. And they give you another example, the Human Genome Project. Human Genome Project was started sometime in the 1990s. And the goal was to sequence all 3 billion and change nucleotide bases in the human genome. And in the early 90s, when this project was started, it was expected to take about 30 years. About 10 years after this project was started in 2003, the Human Genome Project was more or less complete. And it was published. And the knowledge of the human genome shed a lot of light about a lot of human genes, traits, and possible cures for genetic diseases. And there is basically the human genome. It's about 3 billion nucleotides in size. It has about 22,000 genes on it, give or take. That number is getting closer to 20,000 rather than 22,000. The typical human has 46 chromosomes. There's 22 pair of chromosomes and one pair of sex chromosomes. They are not, the sex chromosomes X and Y are not the same thing. And there are only 4 bases that make up DNA. They're adenine, thymine, guanine, and cytosine. Now, some other examples of genetic advances. I'm old enough to remember that when I was in college, and I graduated from college in 1994, it was an impossibility that mammals would be able to be cloned. And then in 1997, it was announced. And that was right when I started graduate school, or soon after I started graduate school, anyway, that Dolly the sheep was cloned in 1997. And Dolly was euthanized in 2004. So this is old news by now. And then in 2002, there was a cat that was cloned by another technique and had a name, carbon copy, or nickname, copycat. And to some extent, cloned plants and animals could benefit farmers. And human cloning, although it is a scientific possibility, it has been legally banned worldwide. Every now and then, someone announces they've cloned humans, or they have edited the human genome to cause disease resistance or something. And yet the scientists that make these announcements refuse to provide the evidence that this is what they indeed have done. And that's a picture of Dolly the sheep. And I believe that is her surrogate mother. And there's Copycat. Other examples, there is a protein that is found in jellyfish. And so certain jellyfish have this gene that makes this green fluorescent protein, which makes bioluminescence, which means under certain conditions, you can see that jellyfish glow green. And you can take this gene and you can put it in any other organism you want to. And you can, for example, put it into a mouse and make the mouse glow. Or you can use this as a marker. And when you clone genes into another organism, you can tie it to this GFP. And it gives you evidence of success. And in the bottom case, they have cloned that gene into gonads of male mosquitoes. And during development, you can see where that particular tissue type is during the development of the mosquito. And so, to some extent, geneticists are going to concern themselves with the expression of genes. And they will be interested in biochemically the ingredients of a cell. How does this DNA store information to make proteins? And then the proteins themselves are the workhorse molecules of the cell. And so DNA stores information, which gets transcribed into RNA, which gets translated into proteins. It's usually the way it goes. And all of the combination of proteins in a cell determine what that cell is going to look like and what type of cell that's going to be. And so geneticists, to some extent, are going to study the relationship between genes and traits. So between genetic information and physical appearance or behavior or something. And so a gene can be defined, more or less, a relatively short segment of DNA that encodes information necessary to make a protein. And you can define a trait as any characteristic that is displayed by an organism. And during the lifespan of the organism, well, genes are going to encode the information that provides these traits. And some examples. And this is just three very simple examples of traits. And for example, you could have eye color, hair texture, height. And then there's other traits that are not as strongly tied. Skin color, for example, is determined by genetics, but it's also determined by the environment. And so there are some traits that are very tightly determined by the genes you have. And there are some traits where it's the genes you have that contribute to that, but it's also the environment that contributes to that trait. And so every living thing is made up of basically four types of molecules. And this is common to anything that is alive. To some extent, it's also common to things that are not alive. For example, viruses are usually not considered to be alive, but you can argue in their favor. And the four basic types of macromolecules that living things have is nucleic acids, proteins, carbohydrates, and lipids. And each of these big groups of molecules plays a certain role in the organism's body. And so basically, we say that nucleic acids, proteins, and carbohydrates are large macromolecules, and they are made up of repeated units of smaller building blocks called monomers. And for example, nucleotides repeatedly make up DNA or RNA. There's a big group of molecules called amino acids. And proteins are made up of amino acid after amino acid after amino acid after amino acid. The lipids are a little bit different. Lipids are not, repeated units of building blocks over and over and over. Different kinds of building blocks interact to form a variety of different lipids. And in eukaryotic organisms, anything with a nucleus, we have a variety of organelles inside the cells. And usually, to keep it simple, we say an organelle is a membrane-bound compartment that has some special function. Let's say, for example, the nucleus. It has a membrane. It's got a nuclear envelope. And inside the nucleus, there are your chromosomes. And at least one of the jobs that your nucleus does is it protects your DNA. It does some other things, but that's basically what it does. And so, in a living thing, the building blocks of DNA are nucleotides, and there's four of them: the A, the G, the T, and the C. To make your DNA fit inside the cell, because your DNA is huge, your chromosomes are gigantic. This DNA has to be combined with proteins to make these big, long structures, chromosomes. And in the case of a eukaryotic organism, your chromosomes are within your nucleus, and they will stay inside that nucleus, except for there are certain stages of cell division when the nucleus breaks down, and therefore the chromosomes are bare at that stage. And then, well, proteins do the work. You have something called the genome. Sum total of all the DNA you have is your genome. And you also have a proteome. Sum total of all the proteins that are in a cell. Every cell in your body is going to be more or less genetically identical. All of the different cell types that you have, and there's a couple of hundred, are going to have different proteomes. And they're going to give the cell its shape and structure. They're going to be involved in transporting things. And by transport, it means transporting things across the cell membrane. There are going to be motor proteins, which move things around inside the cell. They're involved in cell signaling, especially true in a multicellular organism. Every cell you have is identified as to what type of cell it is and which organism it belongs to. And there's a big group of proteins called enzymes that are there to speed up chemical reactions. And in any living thing, the genetic material is deoxyribonucleic acid, which we don't like to say. We say it's DNA. And DNA function, the way it functions, is based upon its structure. And so the individual molecular structure of a DNA molecule allows it to be copied. It allows it to be transcribed into RNA. It ensures that it's copied accurately because A's like to bind to T's and G's like to bind to C's and so on and so forth. And we've described DNA as a polymer of repeated nucleotides, just over and over and over again. Now, as far as I know, DNA has four bases in it. There might be the occasional exotic base, but usually we say there's adenine, thymine, cytosine, and guanine. And the information genetically is stored in the sequence of these A's, T's, C's, and G's. And so it's that sequence of those bases that means something, that allows the organism in question to eventually synthesize proteins that do the work in the cell. Now, all of this should be a review. You had to have biology 1 for science majors to qualify for this class. And so this is a couple of chapters in biology 1. The genetic code is a 3-based sequence. And every 3 bases is called a codon. And every codon means something, or most codons mean something. And these codons specify an amino acid. And when you get the amino acid chain linked together, it becomes flexible. And that polypeptide chain begins to fold. And when it folds up, it adopts a very particular shape. And that very particular shape determines what that polypeptide chain can do. And if the shape is changed, well, the job that polypeptide can do is changed. And for example, they say there's examples of a DNA sequence: ATG, GGC, CTT, AGC. And they say that particular sequence will make the amino acid sequence methionine, glycine, leucine, and serine. And then there's another sequence, and that would make a different amino acid sequence. And you do not have to memorize these sequences. Eventually, you will memorize most of them just based upon repetition. But there is a genetic code table. It has been translated. And if you're ever asked to take a DNA sequence and convert it into amino acid sequence, that table will be provided. And you just have to know how to use it. Now, in living things, all of the DNA is organized into these big structures called chromosomes. And in bacteria, the chromosomes are small and they don't have to be compacted a lot. In eukaryotes, and they say, for example, the average human chromosome contains more than 100 million nucleotides. And that's not entirely true. The biggest ones you have might have 250 million nucleotides or so. And the tiny chromosomes that you might have might contain about 40 million nucleotides or so. And the number of genes on a chromosome, well, it depends on how big the chromosome is. Maybe it's 1,000 different genes. Maybe on little tiny chromosomes, maybe a couple of hundred different genes. But a lot of genes per chromosome. The typical human cell should have 46 chromosomes. 46 chromosomes. And they exist in pairs. And chromosomes are just given numbers. So chromosome number 1 is the biggest, and 2 is the second biggest, and 3 is the third biggest, and so on and so forth. And in the case of mammals, you have chromosomes X and chromosome Y. Chromosome 1 and chromosome 1, one you get from your mother, one you get from your father, they're more or less identical to each other. Same genes are in the same place, etc., etc., etc. Chromosome X and chromosome Y are very different from each other. And you can see there's a big shape difference and big size difference. And the genes on chromosome X and chromosome Y are very different. And it's the combination of X and Y's that determines the sex of the organism. And then non-mammalian organisms might have different sex determination methods. They might have different kinds of chromosomes, or they might depend upon environmental cues to guide the developmental pathway. And so DNA information is utilized through gene expression. And gene expression is simply the use of a gene sequence to determine characteristics of cells and therefore organisms. And it occurs in a few steps. The first step is always transcription, and that is converting the DNA sequence into an RNA sequence. And in the case of a eukaryotic organism, that RNA, which is made in the nucleus, exits the nucleus and goes out into the cytoplasm somewhere. And then that RNA sequence is translated into a polypeptide. That polypeptide then folds up. It makes a specific three-dimensional structure. And it's the particular combination of polypeptides that determines cell type, cell shape, cell function, etc., etc., etc. Now, the relationship between genes and traits is complicated. There is immense variation in between different organisms of the same species. There are different relationships between genes and the proteins they encode and the environment. And the genes have to be transmitted to the offspring in the appropriate dosage. And organisms that are capable of sexual reproduction do this one way. And organisms that are incapable of that do it a different way. And then there's a process called evolution, which is basically genetic change in a population over time. And so a trait, any characteristic that an organism displays, some can be morphological. You can see a difference. Flower color, for example. Some of them are physiological. They affect the underlying chemistry of the organism. Can you metabolize certain things? Can you break down a certain type of sugar or not? Some traits are behavioral. They affect, well, the way you respond to environment. Very often, we like to poke fun at birds at this. Different species of bird have different mating calls. It allows the male and female to identify each other during the appropriate time of the year. And these traits can act on different levels. They can act at the level of the molecule, the exact combination of proteins that a cell and or an organism can make. They can act at the level of the cell. Proteins can alter the function of the structures in the cell. They work at the level of the organism as a whole, which is just traits that are seen in the organism are due to these lower level effects. And they can lead to traits that you see at the level of the population. And a population, and I love this definition of species, all members of the same species living in basically the same area at basically the same time is a population. Basically, all members of a population can reproduce with other members of a population. And the way organisms interact with each other determines that population level of traits. And they give you a very, very oversimplified example. There's a classic experiment that we now know is not exactly as simple as this was laid out. But there's a species of butterfly. And different individual butterflies have different colorations. And you can go ahead and say, at this gene that encodes body color of a butterfly, there are two alleles. And one makes a pigment that is dark, and one makes a pigment that is light. And if the butterflies express a lot of this dark allele, they're dark. And if they don't, well, they're lighter. And at the population level, the coloration of butterfly makes a big difference in their survivability. And this classic example was you have these dark and light butterflies, and they lived in forests. And this was written up in the 1800s, and it was written up in England. And at that time, there was a lot of pollution from coal-fired power plants. And a lot of the pollution was coal dust. And that dark coal dust landed on the plants. And the dark butterflies blended in, but the light butterflies stood out. And so the frequency of light or dark butterflies changed in the population because one was easy to see and get preyed upon, and one was not. And if the situation reversed itself, all that coal dust pollution diffused out. Well, the selective pressures would have changed. And what was once selected against got made more rare versus what was selected for and got more common would change. And this is the classic example. And further research has shown that it's not exactly that simple. There's a little bit more to it than we once thought. Now, inherited differences in traits are caused by genetic variation. And genetic variation means that every member of a population is going to be genetically unique, basically. And you have a couple of easily seen examples. White flowers versus purple flowers. Those were in peas. Black hair versus brown hair. In some cases, the genetic variation within a population can be very, very, very striking. And in some cases, you have members of the same species that, unless you're careful, you would identify as being of two different species because they really don't look anything alike. And you might use the term morph to describe this situation. Very contrasting forms within a single species. For example, same species. If you look on coloration pattern alone, you would not identify these two animals as being members of the same species. So they're morphologically very different from each other. And variation can result in a variety of ways. There are mutations at the level of the gene, and that's going to be a heritable change. Since you inherit your genes from your parents, if that sequence mutates, it changes. You inherit the change. You can have mutations at the level of the chromosome. Chromosomes can break, they can split in half, can be fused together. They can be rearranged in order. You can have changes in the level of chromosome number. And this is usually detrimental. You can lose or gain an entire chromosome. Or you can lose an entire set of chromosomes. So in the case of humans, that means you would have lost all 23 copies of one chromosome, or you have 23 in excess. Changes in chromosome number are usually very detrimental. This is inheritance of an extra chromosome. This is inheriting an entire set of extra chromosomes. Animals don't tolerate these very well. Plants tend to tolerate inheriting entire extra sets of chromosomes. Animals, for whatever reason, don't handle that well. And for most traits, they're not explicable by the genetics alone. A lot of traits result from interaction between genes and environment. And it's got a shorthand. It's a G by E environment. And for example, your diet has an effect on how tall you are, how heavy you are, your intelligence. We now know that your diet has an effect on the functioning of your immune system. We now know that your diet has an effect on what they call your microbiome, all of the little microbes that live on and in you. And the environment might have a major role to play in certain traits. An example, there's a human disease, probably got examples of non-humans with this too. It's called phenylketonuria. And I know that in the United States, every newborn is screened for phenylketonuria. In phenylketonuria, you have inherited a mutation in a gene that encodes an enzyme. And humans need at least one functioning copy of that enzyme. And if you have the disease PKU, you have no functional copies of that enzyme. And there is no real known cure, but there is a treatment. If you have PKU, you don't eat proteins that contain a lot of protein. You don't eat a lot of proteins that contain phenylalanine. And if you have this disease, you can't break down phenylalanine and it builds up. And mostly it affects your central nervous system. And so the treatment is basically from birth. You don't eat phenylalanine. And I have read that once your central nervous system has matured, sometime in your 20s, if you want to, you can go off that special diet and eat things that contain phenylalanine. Because at some stage, your immune system, your nervous system has matured, your brain has reached maturity, and it won't show any massive effects of buildup of phenylalanine. Basically, newborns are screened for PKU at least once, maybe twice. And if it detects positive, you simply don't eat phenylalanine. And without phenylalanine in the diet, development is normal. Okay. During reproduction, genes get passed from one generation to the next. And, this is review, I hope. Gregor Mendel is called the father of genetics. He was working in the 1800s. He described a very, very basic pattern of inheritance. And it turns out that the patterns of inheritance that he elucidated can be explained by how the chromosomes behave during cell division. And some basics. We typically say, to keep it simple, And there are exceptions of this. But we say that species that are capable of sexual reproduction are diploid. Two copies of every chromosome, one from each parent. You call this arrangement, they're in homologs. There exist in homologous pairs. And therefore, again, to keep it simple, since you have two copies of every chromosome, you, in general, have two copies of every gene. There are differences. Genes might multiply. You might have several copies of some genes. But basically, you can say, I have two copies of every chromosome. Therefore, I have two copies of every gene. Now, the copies might be of different alleles. And they say PKU is on chromosome 12. Oh, great. And in the case of mammals, you have chromosome X and Y. And chromosome X and chromosome Y are very different from each other. Very different in size, very different in genetic makeup, all kinds of good things. And so they said, well, in humans, most cells are going to have 46 chromosomes. And to actually put this correctly, you have 22 homologous pairs and one pair of sex chromosomes. That's actually more accurate than saying 23 pair because X and Y are not homologous. And no, that's nitpicky. But anyway. And then if you are capable of sexual reproduction, you are capable of making what they call gametes. And in general, these are referred to as the sperm cells and the egg cells. Male gamete and female gamete. And if the organism in question is diploid, two copies of every chromosome, the gametes are haploid, one copy of every chromosome. And if the machinery that makes the gametes function without error, you should have one copy of every single chromosome in a gamete for a total of 23. And when the two gametes fuse with each other, they go back up to 46. Now, the typical body cell in any organism is what we call a somatic cell. If you're capable of sexual reproduction, somewhere in your body, you have germline cells. And these germline cells make the gametes. And the reason we think, because if you think about it evolutionarily, sexual reproduction is expensive, but it gets maintained because sexual reproduction enhances variability in the population. And so it means that every generation will be slightly different from the previous generation. And it allows populations to, for example, survive new challenges. And I'm sure you all remember a few years ago, there was a new disease that swept around the world. And it just turned out that some humans were genetically resistant to COVID infection, and some were more prone. And basically, it's the lucky ones that were resistant to it, but that resistance allowed the population to survive. Genetic composition of populations is going to change over time. And this is evolution. And biological evolution is usually gradual. It takes a long time for the effects of evolution to be seen. And it exists because of selective forces. Every individual is different. Certain combinations of genes and traits are beneficial, and certain combinations of genes and traits are not. And the combinations that are beneficial tend to increase, and the ones that are not tend to decrease. And this is the family tree of the modern horse. And over a period of a few tens of millions of years, well, there were very important changes. Body size got larger, toes got reduced in number until you have the modern horse, which was one toe on the ground. Teeth and jaws changed. Long time ago, basically, animals were fruit eating, and fruit has a lot compared to grasses. They're a lot more calorie rich. And if you have to graze, you're eating a lot of grass. And the animal can't digest all of this plant material. And so it has to eat a lot in order to get enough calories to survive. And so there's a few fields of genetics. And they said, well, there are transmission genetics, molecular genetics, population genetics. And if you wanted to split hairs, you could add a few more. Now, primarily, a geneticist is going to use a variety of model organisms to figure out problems that they're trying to solve. And basically, there's a reason. One of the reasons is basically any living thing has DNA in it and it behaves by the same rules as any other living thing. Another reason is in your course study, you are likely to kill a lot of this population of your organism. And the thing is, if you kill a whole bunch of bacteria, no one really cares. If you start off working with humans, and in the course of your research, you destroy a lot of your population, well, people are going to begin to care about that. But you can work with simpler organisms that are easy to grow in lab or fast, and you can extrapolate from one organism to another. And some of the model organisms, this is a bacteria, this is a little nematode worm, here's yeast, fruit flies. If you're working with plants, you are going to work with a lot of Arabidopsis. If you're working with animals, you work with a lot of mice, etc. Now, transmission genetics explores patterns of inheritance. And it just is going to see how traits get passed from one generation to the next. The early groundwork of transmission genetics was laid down in the 1860s. And the vocabulary that is used in transmission genetics has changed. Gregor Mendel didn't use the term gene. He used the term inheritance factor. Nowadays, we say these things are genes. And Mendel used specific genetic crosses to analyze inheritance patterns. He would select the parents, make the offspring, and see what the offspring looked like. And there are some examples. And I have a formula. It's written on my desk, taped to the desk here. How can two brown-eyed parents have a blue-eyed child? Why do tall parents tend to have children that are tall, but not always? Why do two tall parents have children that might be tall but not as tall as they are? And the next few chapters is going to cover questions in transmission genetics. And then you have genetics at the biochemical level, looking at the structure of DNA and RNA and protein. And at the start, molecular geneticists will start with basically a transmission genetics approach to figuring this out. But at the heart of it, molecular genetics is really want to study genes that are mutated. And there's two basic kinds of mutation. There's gain of function mutations, and there's loss of function mutation. And loss of function mutation means you have a mutation where the organism that has that mutation does not make a functional protein of some sort. And then molecular genetics, what is it good for? Well, it has, well, interrelationships with biochemistry, biophysics, cell biology, things like that. Molecular genetics can be used to shed light, to illustrate genetics at the transmission level and the population level. And at least currently, these molecular technologies are computer based, and it allows you to analyze things very rapidly. And you can apply the data that you might uncover to all kinds of different fields. And then you have population genetics. And population genetics is what I did. And every allele of every gene in a population exists in different frequencies. And Darwin was one of the very first scientists that began to apply these different frequencies to theories of evolution. And there are all kinds of different mathematical equations and theories trying to explain why certain alleles are common, why they're prevalent, and why other alleles are more rare. And at the basis of population genetics, knowing allele frequencies is very important. And it allows you to make some predictions about what the population might look like in the future. And then genetics is considered to be an experimental science. And you might use hypothesis testing, which is use of the scientific method, or what they call discovery-based science. And I happen to know, for example, the author of this textbook, Robert Brooker, is a genomicist. All he does is want to collect data about genomic structure in different organisms. He doesn't really want to explain it. He doesn't want to say why it's there. He just wants to find it. And he wants to publish it. And maybe at some later date, someone else will come along and find his data and develop a hypothesis, develop an idea about why this exists, and begin to test that hypothesis based upon his discovery. Now, this textbook usually chops experiments into a few different steps. Background explanation, development of a proposed explanation of why you're seeing what you're seeing, designing an experiment, Your experiment should give you data. And then you have to analyze that data with statistics. And then you have to form some kind of explanation of that data. And it's a good idea to come up with, you know, your own skills to make hypotheses or design experiments, which is not the world's easiest thing to do. And interpret data, which again is not the world's easiest thing to do, but it's a good idea to begin to practice it. Now, this textbook says it uses this strategy. And so it advises you to use steps over and over and over again. Look for key terms, bold print words. It says to make a drawing, perhaps give you a picture on what's going on. Make predictions. Compare things together. Realize that structure and function are related. Make these hypotheses, design an experiment, learn how to basically analyze data. And we use some very, very rudimentary calculation skills. Now, hopefully, the presentation that you just saw, which was from the fourth edition, is not too different from anything that might be in the 2025 publication of the book. Because, like I said last week, they have the same cover. Page numbers are basically the same. I did have to make changes into the reading, so the page numbers are different. And so something in the 2025 publication is different from something in the fourth edition. I just haven't found anything screaming that there's a huge difference. And remember that the exam schedule that I have is hypothetical. It could change. Some chapters are long. Some chapters are a lot shorter. And it is nearly 2:30. That's long enough. And again, I'm going to end this. If you come up with any questions, email me. Because I usually end it too fast to answer any questions that people come up with, like now. But you can email me and I'll do my best to respond.